on to part 2, folks. The second half of glycolysis starts with the enzyme glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. You may already know of GAPDH if you've ever run a Western blot with GAPDH as the control. So this enzyme takes our molecule of GAP and converts it to 1,3-bisphosphoglycerate, or 1,3-BPG. You'll also see, though, folks, that we use an NAD+, and it's converted to NADH. This tells you that we have a redox reaction happening. So this enzyme does both oxidation of GAP and simultaneous phosphorylation. You can see that we go from this terminal aldehyde that has a double bond to oxygen and a bond to hydrogen to now this phosphoanhydride looking bond where we have carbon double bonded to oxygen and then also bonded to oxygen and phosphate. Step 6, we have the formation of the first high energy bond called an acyl phosphate. I think this mechanism is super cool and we should have a basic understanding of it. I think it's cool because this is the first time really that we've seen the redox molecules in action. So let's walk our way through this mechanism together. So we're starting with this catalytic site. We have a thiol, we have a base, we have NAD+. In comes our substrate, GAP. Recall that the reaction is happening at the terminal aldehyde. The base grabs a proton from the thiol. SH, not a very good nucleophile. S-, much better nucleophile. For that sulfur then to attack the carbonyl carbon. That gets us up here, where our base is protonated and positively charged, and the sulfur group has formed a transient covalent bond with our substrate. This is the point I want to point out because it's cool. Okay, carbon can only make 4 bonds at a time. So when we collapse back down the negatively charged tetrahedral intermediate, this hydrogen breaks off as hydride. So it's an H with 2 electrons and a negative charge. And that hydride is a strong nucleophile, the strongest really, that attacks the NAD while performing a redox reaction. Cool stuff. The rest of this reaction should be fairly easy for you to follow. So I think this is super interesting because the reaction at the hydride is new for us. Okay, folks, what did I say? We have the formation of the first high energy bond in step 6. What's the point of forming high energy bonds? Well, to harvest the energy from that bond. So as soon as we see a high energy bond formed, in the next step, we're going to harvest that energy to create ATP and recognize that we're really creating 2 ATP because we have 2 molecules of GAP we're starting with. This step is performed by phosphoglycerate kinase, where we take 1,3-BPG and an ADP, and we end up with 3-PG, so we've grabbed one of the phosphates off, and we have synthesized ATP. Sorry, my face is covering that. The mechanism is super similar to hexokinase, it's just kind of in reverse. You can even see, folks, kind of the structure-function relationship here because this enzyme shape with the open Pac-Man mouth before it binds substrate is very similar to what we see in hexokinase. So step 7, we form our first ATP. As an aside, folks, reactions 6 and 7 are energetically coupled. Reaction number 6, I didn't show you this, but it's actually not favorable, it's not spontaneous. Except reaction number 7 is favorable enough, it releases enough energy to pull reaction 6 through. So this is what we called a coupled reaction that we talked about last time we were together. Additionally, folks, I want to make a distinction between the synthesis of ATP we're seeing in glycolysis with the synthesis we will see of ATP through ATP synthase after the electron transport chain. This type of ATP synthesis is called substrate level phosphorylation. And I generally think of substrate level phosphorylation as the synthesis of ATP from the transfer of a phosphate from some metabolite, some substrate, for example. We transferred a phosphate from 1,3-BPG to ADP to make ATP. That is unique to substrate level phosphorylation. We will see oxidative phosphorylation, the synthesis of ATP through oxidation and reduction, later on. Up next, folks, the enzyme phosphoglycerate mutase. So this enzyme takes 3-PG and isomerizes it to 2-PG. So we're just popping off the phosphate from the third carbon and putting it on the second carbon instead. I think this enzyme is cool because it actually contains a phosphorylated histidine in its active site that is a part of the mechanism. So basically what's happening is the substrate has a phosphate and the enzyme has a phosphate, and they just kind of trade with each other. One additional interesting thing to point out for this enzyme is that the intermediate of the reaction, so like the halfway point of its mechanism, we have the formation of a molecule called 2,3-BPG, which previously in this course I just kind of simplified as BPG. Where have we seen this before? When we discussed hemoglobin. So a cool link back to something that we've already known. Here is the intermediate shown to you on the slide, folks. So you can see how basically what was happening was this trade where a phosphate was given from the histidine to carbon number 2, and you can imagine now how the substrate will basically just give a phosphate back. Why do we need step 8? Why do we need to move this phosphate from the third carbon to the second? Basically, we can't form a high energy bond if we have this phosphate here. We need to get this phosphate closer to what's happening up here to form this phosphoanhydride-like situation. So we move the phosphate so that we can form a high energy intermediate, which is what we do in the very next step. The second to last step of glycolysis, we form our second high energy molecule through a dehydration. I have no idea why Zoom just did balloons over my picture. That was super weird. So we're taking 2-PG and converting it to phosphoenol pyruvate, or PEP. Okay? PEP is this molecule with this extensive, almost conjugated system, right? And this contains a high energy bond. Something interesting about this enzyme, folks, but not just this enzyme, it's really any enzyme that requires magnesium, is it can be competitively inhibited by fluoride ion. Right? Fluoride ion, oops. You know, sometimes it's been a long time since I've thought about the periodic table. That was not the correct abbreviation for fluorine. Okay, so the fluoride ion, folks, is able to complex with magnesium. What do I mean by that? It performs a favorable non-covalent interaction where they kind of just bond, interact with each other non-covalently. So it's able to kind of grab magnesium. There are a lot of enzymes in glycolysis that require magnesium to do charge stabilization. So I thought that was interesting. Finally, folks, in one step, if we form a high energy intermediate, in the next step, we harvest that energy to synthesize ATP. So that brings us to the last enzyme of glycolysis, pyruvate kinase, that converts PEP into, finally, pyruvate. Along the way, we cleave the high energy bond to convert ADP into ATP. This is the third rate determining step of glycolysis. And we see another substrate level phosphorylation and formation of 2 more ATP, because remember, this reaction is happening twice for each molecule of glucose. The interesting thing about this reaction, folks, is that it's actually not really the hydrolysis of this phosphate that's giving us most of the energy that's released. It's the tautomerization from enol to keto of the intermediate that provides most of the energy. And I show that here on the slide. So it's not really this hydrolysis that's the biggest deal. Yes, it releases energy. But look, if you wanted to be convinced that the keto form of a molecule is more stable than the enol, this is the reaction to convince you. Because simply by going from enol to keto, we release an additional almost 50 kilojoules per mole. Some other notes while we're here on the slide, folks. Look how much charge stability is happening with this enzyme. The enzyme has a magnesium and a potassium here to help stabilize all of the negative charge on the substrates. Cool stuff. And that is the second half of glycolysis. We saw 2-GAP converted into 2-pyruvate. Along the way, we made 2-NADH and 2-ATP. So we have a net production of 2-NADH and 2-ATP, right? Take 1 molecule of glucose in, 2 molecules of pyruvate out, 2 molecules of NADH, 2 molecules of ATP. And all of this is happening in the cytosol. We learned a lot today. We walked our way through each of the 10 enzymes. We don't need to memorize them all. That's what the handout is for. But we should understand something about each of them. Now, as a big picture looking forward, right, we should be able to predict how changing parts of the pathway will affect flux. So into the future, we'll be thinking a lot about flux through metabolic pathways, and we'll learn how to link the pathways together. See you next time, folks.